So it is my pleasure to introduce Professor Kevin Talbot, the Professor and Head of Department in the Motor Neuron Biology at Oxford, UK. And as you do with your children, you try to expose them to people's lives and what they do. And I was trying to explain to my 12-year-old what Professor Talbot does. And I went through the fact that he was a clinician, he was a translational researcher, he was a head of department, he administered, and he was, you know, he was sitting there and I could see that he was taking it all in. And then he sort of, the acknowledgement occurred. So his answer to that was, so he's got a big brain. And I said, and then he followed it with, is it bigger than Professor Kernan's? The answer to which was, I'm not sure. But anyway, I won't go through the list of his achievements, but it's great to have him back in Australia. Luckily for us, he's staying for a period of time, so we'll be able to squeeze all the juice we can out of him. Thank you, Kevin.
Well, thank you very much. And I certainly cannot follow that wonderful talk from Phil, which I hope, you know, sends everyone away thinking hard. I've got to try and keep this as non-technical as possible so that I can talk to everybody in the room. So let's see how we get on. I mean, we need to understand, and what I do in clinic, as my colleagues will do as well, is you talk about genes and cells, and you realize very rapidly that most people don't really know what you're talking about. So let's just be clear for everyone in the room that genes are the template for RNA. RNA produces protein, and proteins are best thought of as like a house. They either provide the bricks and mortar, the structure of the house and the building, or they make it work. So they might be things that, you know, are involved in wiring, plumbing, and energy production in the house. We have 20,000 genes, which is slightly fewer than a mouse, but we can produce from that an unknown number of proteins. No one's really sure, it's probably millions. And so it's extremely complicated. So when we talk about genetic disorders, we're talking about a gene being altered, leading to a protein being altered or absent, which changes the function of a cell. So when we sit in clinic and we use the word familial ALS, which I think increasingly is a blurred boundary between familial and sporadic, what we're saying is that we've talked to the patient and we've found a family history, like the one here. And that means that there are people in each generation who also have motor neuron disease or a related condition. And then we go looking for genes. And then we find a gene mutation like this one here, which is SOD1I114T. But I think the good news in terms of what we're trying to do here is that these patients look exactly the same as the ones who don't have mutations. So there's something going on which is in common, whatever the underlying cause. But the other thing is that, and this is things that Amar emphasized at the beginning of this meeting, familial does not mean, it's not the same thing as genetic. So there are now 40 genes or varying degrees of credibility as causing ALS or being associated with ALS. And of those, SOD1, TAR-DDP, FUS, and C9orf72 are the commonest. But if you look at populations of people in your clinic who carry these mutations, so like lots of people, we've sequenced, you know, a couple of hundred people to try and understand the genetic architecture of the population of patients we're bringing into trials and studies, what you find is there are more people in your clinic who don't have a family history than do have a family history. So familial and genetic are not the same thing. And there's lots of studies that have shown that. And the other thing is that it depends where you are in the world. So if you're in the Far East, you'll have a different genetic profile to the one you have in Australia and other countries where there's a large population of European genetic heritage. This is work that a review that Thanuja wrote when she was with us. And then there's the issue of reduced penetrance. So the penetrance means what's the probability if you carry a gene mutation that you'll develop a disease. And you really need to understand penetrance in these disorders if you want to understand what turns somebody carrying a genetic variant into somebody with a disease. So having the genetic variant is not a sufficient condition for the disease. It's there from conception. So it's not toxic actually at all. It's tolerated for decades and decades. So we sit in these meetings, we talk about gene toxicity, and we've got to be very careful what we think we mean by that, because that's not what we're seeing in patients. We're seeing people tolerating genetic mutations until we see the intersection of aging. And actually, it turns out, this is work done by Andrew Douglas in our group, who's a clinical geneticist embedded in our group. So he looked at all the available databases of sequence data and looked across the 4 commonest genes. And for each of them, there are more people in those databases with predicted pathogenic variants than seem to be presenting to our clinic. So all of these genes have apparently reduced penetrance. And the dramatic one is C9orf72, where there may be, up to 3 or 4 times the numbers. If you look at UK Biobank, for example, which is 500,000 people, and we've looked at the number of C9orf72 hexanuclide repeat expansion carriers in that cohort, and there are far more than would ever be predicted to present on current data to our clinics. So that's the problem, and it's something, I'm stating that because later on we come back to what we do with that information and how we might need to use it. Okay, so you can treat these genetic defects by replacing a gene if it's missing. That's a straightforward gene therapy. You can package it in a virus and you can give it. So spinal muscular atrophy, for example, which you'll all be aware of, is a triumph of molecular therapeutics. It's really changed our view of what's possible to take a completely untreatable condition and prevent it effectively in as much as we know at the moment. So you can replace something, and for MND, that's largely not what we're going to be doing because that's mostly true of rare recessive disorders. There may well be some examples in time. Or you can neutralize this harmful variant. And this is a missense mutation, so it changes the code and causes a protein that is either going to be degraded or misfolded or behave differently. It's going to acquire a different function under certain circumstances. And so in those contexts, you are going to be using something like antisense, or ultimately you might be using genome editing. So antisense therapy briefly is, at its most commonly used form, engages a target RNA and recruits in the enzyme that degrades it. So it simply degrades the messenger RNA, which contains the variant, hopefully, but also it may contain the normal. It will do the same for the normal gene unless you have an allele-specific oligonucleotide. So an oligonucleotide is a small piece of nucleic acid, DNA or RNA, which binds to the RNA and therefore will prevent the production of that protein, which is going to cause misbehavior. Exon skipping is a way of taking the messenger RNA, which contains bits that contribute to the protein ultimately and bits that don't, and the bits that don't are spliced out. And exon skipping can engage that messenger RNA and recruit in the machinery that includes, or maybe even excludes, particular exons. And that actually is in its infancy in a way, but ultimately it may be a therapy when you want to alter a gene that may just be, contributing to the pathogenesis of a disease. So I think there'll be a lot more exon skipping type stuff going on. Okay, so let's get straight to, as we haven't got much time, straight to the results that we are aware of for SOD1 and for C9orf72 and for FUS. So this study, you've seen these graphs already, is probably the single most important development in ALS in my working lifetime because, I think I have occasionally sat in clinic and wondered whether ALS isn't a treatable condition, and once it starts, whatever pathological cascade is there, that it might just continue whatever you do. And that was a formal possibility. But above all, what this tells me is that that's not true, that you can stop the progression of the disease and maybe the body can be encouraged to reverse that condition. So I think that's a landmark and gets over the sort of neurotic concern that maybe this is not so treatable. And that's, I think, conceptually why it's so important. Obviously, it's important to those people carrying the mutations who might benefit. So in this study, it shows pretty clearly that neurofilaments are reduced prior to statistically significant clinical benefit. And that's also been a very helpful kind of development because it's allowed us to focus on neurofilaments as a potential tool for deciding whether a drug is engaging the disease in a much quicker time frame than if we wait simply for outcome measures like function. So that's now been approved in the United States and in Europe. And we're arguing in the UK, as often is the case, to get it approved there. So what about the real world data? So that's a trial, and that establishes that the drug is doing something. So there are a number of studies which are looking at the real world activity of this drug. And of course, not surprisingly, it's much more complicated. So you'll see people who clearly are stabilizing here. There are people who have declining neurofilaments, those who maybe don't have such a decline, or it fluctuates. There's quite a lot of mess and sort of noise going on there. And I think that's just the real world. That's why you need to do trials. But there are people in this particular Italian group of about 17 patients who continue to decline despite taking tofersen. So that may suggest that they had advanced disease. It might suggest that there are responders and non-responders. It might suggest that there are person-specific factors in terms of the delivery of this drug, in terms of its pharmacokinetics and its engagement with the target. So all of these things need to be understood in much more detail. We need a redefinition of the natural history of SOD1 ALS in the treated context to understand exactly how generally applicable this is. And along the way will come other therapies. So there are several now talking about recruiting, and it raises another issue. So who in your clinic is going to go into a placebo-controlled trial of a different type of antisense or a virally delivered therapy if tofersen is available? I think this is one of the challenges for the whole field as to how on earth we're going to negotiate that. So FUS, those who were at the meeting in Basel will have seen an incredibly moving presentation where a young person seemed to have a really dramatic benefit from this antisense against FUS. FUS is the worst possible disease. Young people, very aggressive. And this antisense developed at Columbia University seemed to be having an effect. And it's now in clinical trials run by Ionis, and hopefully the results will be positive. Now, C9orf72 is the commonest genetic mutation. So I only put this up here not to go through the biology of C9orf72, simply to remind you that it's complicated. There are multiple mechanisms, which is important to bear in mind. So this is a paper that's hot off the press in the Lancet Neurology this September, showing the clinical trial of Biogen's antisense therapy. And you can see that the you can see that the treated group declined, and possibly even at a higher, faster or a more severe decline than the untreated group. This drug didn't work, and it may have been toxic. The neurofilament levels also seem to increase as well. So something not right about the Biogen drug. Is it the chemistry? Is it something else? The WAVE study. So we were a center in the WAVE study, and this was very interesting. It's a completely different approach with a kind of novel way of producing a stereo pure antisense oligonucleotide. So the preclinical data was very interesting. And this also didn't work. Not only that, it seemed to be toxic. So we don't have any published data. All I have is the data from 2 patients that were in our center. And you can see here that in this patient, there was no reduction in poly-GP, and the neurofilament stayed pretty much the same, and that patient had placebo. And in this patient, the neurofilaments went shooting up, and the poly-GP did go down, but not very dramatically. So this appeared to be quite toxic, and that patient did undergo, unfortunately, quite a precipitous decline, and that seemed to be the outcome of the study. So they're not continuing the development of that drug, and Biogen are not continuing the development of their antisense, which is hugely, hugely disappointing, because, you know, why did these trials fail? So that's a debate. Was the chemistry toxic? Well, 2 different types of chemistry. So, you know, I don't know if that's true, but maybe it wasn't really engaging the target, despite the claims that polydipeptides were reduced. I mean, as you can see from the one patient that we had, it wasn't especially dramatic. Maybe it didn't really engage the target that's relevant to the disease process. And by that, I mean, for example, is the reverse strand antisense that generates the dipeptides the target. So here is some unpublished data from my group showing that if you use a technique called RNA scope, you can see that the antisense, so the reverse strand RNA, is much more abundant in iPSCs than the forward strand. And Pam Shaw's group previously showed that neuropathologically. So the reverse strand may be toxic, and Jeff Rothstein has done a study recently which might support the idea that antisense, the terminology is confusing here, ASOs against the reverse strand may be actually required to have an effect. So the challenges here are that we are going to have therapies ultimately that can be given in early disease or even before disease begins. For that to be the case, for the latter, they have to be totally safe. If we want to treat people who are perfectly well, particularly when the penetrance of these disorders is not 100%, we could be treating people who may never develop the disease in the way that we treat people with antihypertensives and lipid-lowering drugs who may never have a heart attack, because we know their probability is declined. These drugs are safe, it's straightforward. So we can't give something that's toxic to people who are perfectly well. We need to know that these things really get where we think they get to, and we need no off-target effects, because that ultimately will become a problem. So again, if you want to treat a disorder like TDP-43, for example, which we all think is relevant to the disease, then the mutation, those carrying mutations, which there are many, you can't comfortably, depending on your viewpoint, use an allele non-specific version of an antisense. So reducing TDP-43 is a toxic thing to do, and it seems beyond the reach of current antisense approaches, at least to me. You need ultimately to individualize treatment. There are lots of people out there with rare genetic mutations and rare genes. Who is going to fund the development of those? And you need ultimately to have an economic model that allows you to treat people before the disease occurs in large numbers. There are ways in which you can improve the delivery of antisense by packaging them up into nanoparticles of one kind or another, by conjugating peptides. These are technical problems that will dramatically improve their delivery. Focused ultrasound, somebody mentioned earlier on today, may be one way forward. You can alter the chemistry, which might reduce toxicity and might reduce or might increase the level of target engagement. So all of these are technical things that will rapidly develop over the next few years, not conceptual problems. I think we have a concept here. What we need is technological improvement. So genome editing is conceptually the most attractive thing to do. Let's just remove the genetic error that's causing the problem. And of course, the same things apply. You have to do that with complete safety. And you can do it in a dish. So we've done that, as lots of other people have. Doing it in a human requires 100% safety. It requires efficiency that allows you to do it in a non-dividing tissue, the brain. And ultimately, you decide on the timing. Are you going to do this in infancy when people are 20, 30, 40? These are unanswered questions. Okay, so that gets to the point of when does motor neuron disease even begin? It's an open question. We know that Parkinson's and Alzheimer's begin decades before people present. What about motor neuron disease? We simply don't know. But let's assume there is a preclinical period measured in years in which there's a biological process going on that does not lead to loss of function, which would make the patient present to a neurologist. That's what I would call the preclinical landscape. And there's compensation going on, there is reserve capacity going on, but there's some biology that you might want to try and understand. And if you can understand that, it will be the way to kind of get to the issue of how you treat people. So if you understand that biological landscape, perhaps you can understand who it is in those C9 or 72 group who will never develop the disease or who will, and those who will, at what point are they developing the disease, so that you can institute your treatment. And you know, you're going to have to come up with something that has an economic model, treating people with antisense, you know, for decades who are well, doesn't really feel like something we'll be doing. So we have set up a study called ACORN, a C9orf72 national study, and in which we are collecting as many people as we can from the UK who are the relatives of people with the mutation. We'll get about 400 in a few years. We've already got 50 in the first 6 months. And then they'll be research accessible to anyone who has the right ideas. And we'll be doing longitudinal blood, CSF, MRI, and magnetoencephalography to look at the system-wide changes that are occurring in the brain, hopefully some years before the disease may actually present. We've got remote monitoring of physical activity and cognitive function using iPads. So it's quite a well-funded, well-organized study. And, over the years, we hope that will actually produce some of the data that will allow us to know when we can apply these therapies. So NUJA is setting up something in parallel here in Australia. Luckily, the A, fits perfectly well with Australia as well. So it's called ACORN here. GenFi is a frontotemporal dementia initiative, which has been going for a number of years, which we're part of, and we are aligned with them in terms of protocols and data sharing and sample sharing. So we'll try and answer some of these questions. But something that Phil said earlier is the key here. This is a partnership between us and people living with motor neuron disease, affected by motor neuron disease because they're at risk and their family is affected. So, there is no question that people are highly motivated to participate in this. And we didn't just sort of make these things up. People sent us these messages saying how meaningful they found it to actually belong to this long-term project, which we're doing together. And without patients, of course, we will get nowhere. So it's fundamental. So we set up a thing called Fathom, which is Families for the Treatment of Hereditary MND, where we meet, it was every 2 years, we may make it annually. And they come and they listen to us talk about research update and progress, and we engage them in that way, and all the videos are online. So this is a way of building a community of people who together are going to try and address these problems. And ultimately, what's successful therapy going to look like? Well, what I've been talking about is precision medicine. It's about how you target a specific molecular defect. But I think even in people in whom you do that, you may ultimately still need other approaches. So combinatorial therapy with drugs acting on different pathways, and ultimately prevention in order to really make a difference, because rewiring the nervous system once it's undergone change is at the moment a distant prospect. So even those people who have gene-based therapies, I think, will also be in need of other therapies to maintain the health of their nervous system going forward. I haven't really shown you much data from my lab, but this is just to show you a large group of people in Oxford who are involved in various things. My colleague Martin Turner, is somebody who's doing all the imaging and a lot of the biomarker work. Alex Thompson is the PI of ACORN, and we've got lots of people engaged and involved in it. So thanks very much for listening.
Thank you, Kevin. We do have time for one or two questions before we move to the next session. If anybody's got a burning desire over there.
Hello. Thanks for the great talk. So I had a question. You talked a lot about compensatory mechanisms, and a lot of what is done to look at TDP43 biology is, overexpressed and maybe less physiological. And then there are knock-in models and there are iPS cells where often we're working to try to drive a pathological signature. Do you think there's also a possibility to be using those more to look at how compensatory mechanisms are happening, where you're not getting that pathological signature, but after a certain length of time, you might be able to see those protective mechanisms that we have in those individuals who are holding off getting disease?
Yeah, absolutely, I do. And I think that when my postdocs or students say there's no difference between the cells in a dish, I'm actually okay with that, because if they're falling over and dying, then I'm simply going to be picking up the biology of the cell death. And that's not what I want to know. I want to know why those cells are alive in a dish with a mutation like C9orf72. What compensation is going on? It's a much more difficult kind of question to approach, because you need to search a bit harder. But I'm absolutely sure that's what we should be doing, rather than overexpressing things to make them toxic, force phenotypes, which may have some relationship to the disease, but maybe you're forcing the end stage of a disease into your model, which is not going to be therapeutically tractable. So I do believe we need to understand how you can live for 60 years with these mutations and develop your brain normally, or maybe it's even a better brain.
If there are no other questions, no. Oh yes, whoa, there, whoa, just got in, just by this much.
Hi, thank you so much for that. I'm a representative from a C9 positive family. And I guess I just have a question which may not be answerable, but one I've pondered a little bit, and that is CRISPR ever likely to be affordable for the average person. I totally get what you're saying about at what age would you possibly give it? How would we sort of, once we have the technology and it's safe, make our case to have this as something we could do?
Yeah, so I think it's true of all of these therapies. You know, we're in the foothills here, where antisense has been developed by large pharma at considerable cost. But all of that is going to hopefully become a little bit more tractable. So there are multiple groups around the world, and there's one group in Oxford who are trying to address this problem of how you take an individual with a rare disease and you do a bespoke treatment. They're mostly focusing on children with rare metabolic disorders. How can you take that child and develop an antisense in 2 months and get it into that patient? And then what we need to do is we need to partner with pharma and produce a kind of social contract which says this needs to be affordable. I mean, pharma are mandated to make profit for their shareholders. That's why they behave they do. It doesn't really mean it costs that much. So you need a social contract where everyone actually agrees that this is in everyone's interest to get it out there at an affordable cost. Maybe I'm being optimistic, but I think that's what we'll do.